He first joined this conference in 2019. It was an in-person conference in this very room, and he was super interested in joining it and learning about what we were doing, but he sounded kind of confused. He was saying, oh, that's cool, but I wasn't sure that he's really understanding it. So I have to confess, I did have some misgivings, but he seemed to bring a lot of energy and enthusiasm. So I decided to give it a try. Let's see if we can work with Andrew. And I gave him this really ambitious, bold, wacky idea. Let's go and redesign entirely sample prep and do something that doesn't look like anything has been done before. We're going to abandon tubes, we're going to abandon wells, we're going to do things in a different flexible format, and we're going to try to push the limits so that we can increase the throughput. And maybe I was just taking advantage of his naivete and not knowing much, but he agreed, he signed in. And he's driven this project with a tremendous amount of energy and creativity and enthusiasm. It's just been a blast working with Andrew. Whatever initial misgivings I might have had about the enthusiastic, bright-eyed student who is not fully understanding what we're doing, now all of that is gone. He is the master. He can make cell-in-one sing, and he can make it work day in and day out. So it's been a wonderful, wonderful journey to work with Andrew. And I'll welcome him now to come and tell us about the sample preparation that he's developed, taking a lot of the lead on all the things that he's going to tell you about. And please don't be shy to ask questions.
All right, can everyone hear me? So thanks, Nikolai, for that introduction. And thanks, Josh, for the great presentation. I've talked with Josh a lot over the past 2 years since I started working with the instrument. Almost entirely because I've broken something and I called him in a panic to try and fix it. But yesterday I got to work with him and automate some of the last parts of our protocol, and you'll be hearing a little bit about that, and it was really exciting. And he's an awesome engineer. A great guy if you have any questions about the capabilities of Cell-in-One. So, like I said, my name's Andrew. I'm finishing up my second year as a graduate student in Slavov lab. And today I'm going to be telling you guys about the new droplet sample preparation method that we've developed that I'm really excited to share with you guys. So let's get started. So as always, to start with some motivation for why we're interested in doing single-cell proteomics by mass spectrometry. The biological samples we're interested in analyzing come in a wide array of complexity. And we know that in many of these such cases, taking bulk measurements or population averages can obscure a lot of the interesting biological variability that comes from these samples being comprised of cells of different types and different cell states. For example, at kind of the high complexity end of the spectrum, we know primary tumor samples are incredibly heterogeneous and may consist of not only several different genetic variants of cancer, healthy tissues, but also immune cells that have infiltrated the tumor microenvironment, and other cell types that all play a complex role in the progression of this disease. Kind of in the middle, we have a protocol that you might want to pursue for differentiating stem cells, and you might have a target of interest in mind. But along the way, these cells are going to undergo various branch trajectories and bifurcation events spinning off into unwanted products. And to fully understand these continuous transitions in cell states, often we need single cell resolution to understand the dynamics that are driving this differentiation. And at the low complexity end of the spectrum, you have a dish of the clonal population of cells where you think there's probably not too much interesting biological variability going on here at the protein levels from cell to cell. But in fact, even in cases such as these, processes such as the cell cycle produce meaningful and interesting biological variability that goes on from cell to cell. And here you can see some fluorescent images of just a HeLa cell undergoing cell division. And what you can see from this picture is that there are...
Your mic may not be working. Is it turned on?
Oh, maybe I didn't do that. I think people couldn't hear me.
It's off. All right. People couldn't hear you, but now they should.
Oh, should I start over? Oh, okay. Okay. All right. Hopefully everyone was able to hear me all right. Sorry about that. I probably should have realized. So like I was saying, here we have some fluorescent images of a cell undergoing, a HeLa cell undergoing cell division. And what you can clearly take away from these pictures is that there's coordinated changes in protein abundance going on as the cell is proceeding throughout the cell cycle. But unfortunately, with fluorescent measurement techniques, we're only able to measure just a handful of proteins simultaneously. And fortunately, methods such as mass spectrometry allow us to quantify hundreds to thousands of proteins per sample. But again, if you're interested in analyzing a process like the cell cycle on the bulk level, you might be limited by the proper time resolution or introduce artifacts that confound your biological interpretation. So single cell resolution gives us a more unbiased way to sample the wide array of cell states as cells undergo continuous shifts in protein abundances, such as the cell is dividing. So great, let's do single cell mass spectrometry analysis. But as you may expect, there's several challenges to doing single cell mass spec in general, from the measurement side to the sample preparation. And so the sample preparation is going to be mostly what we're focused on today. And so just to give you an idea of what some of these challenges look like, you could imagine preparing a single cell in your average microcentrifuge tube. And just to give you an idea of what a scale is like for a single cell to be contained in such a volume, this would be roughly like if you decided to cage your pet hamster inside of one of the world's tallest skyscrapers. And so you might be thinking, okay, well, the hamster has a big cage. Why is this such a big problem? And this brings me to two important aspects of the sample preparation reaction. And one is the concentration of reagents. We need to make sure that we're adding enough reagents to our samples so that our reactions proceed in a timely manner. And so for analogy, you can imagine if you went into this building to look for your pet, it might take you an incredibly long time to find it on your own. Conversely, you could hire a team of people to search each floor and you'd probably find your pet a lot sooner. But this brings me to the next really important point of the sample preparation reaction, which is the reagent of the ratio of reagents to substrates. And so we really don't want to be adding so much reagents to our sample preparation that it makes it difficult to measure the peptides and proteins that we're interested in measuring. To extend the analogy, you'd imagine it'd take a much shorter time to just ask one person if they had found your pet than to ask a whole crowd of people who had been searching. So this brings us to the next question of how can we reduce our sample preparation volumes so that we're able to maintain a high concentration and allow our reactions to proceed quickly, but also not introduce so many reagents that it makes the analysis challenging. And so this very problem has motivated several different sample preparation techniques. I'm just going to briefly mention a few here. One, Mpop, which we've talked about a little bit, that Harrison Speck developed, that uses volumes of about 1 microliter to prepare single cells in a 96 or 384 plate. And also another familiar method, nanopots, which uses microfabricated chips at volumes on the nanoliter scale that has allowed really sensitive analyses such as label-free single cell proteomics. But we're not just interested in preparing our samples well, we're also interested in preparing many samples so that we're able to sample the wide array of diversity that we might be finding in the samples that we want to analyze. And briefly, I'm just going to talk about some of the advances in single cell RNA sequencing. Methods such as DropSeq and InDrops and 10x genomics really enabled a rapid increase in the scale of sample preparation as well as quality. And this allowed for all sorts of analysis that were previously not possible at the scale that were being analyzed. And so we took a lot of inspiration from this high throughput sample preparation, and we kept this in mind when looking to develop our own sample preparation method. And so just to summarize on what some of the desirables were when we were setting out to kind of redesign our sample prep. One, as I mentioned, we wanted to decrease our sample volumes, not only to add less reagents to our samples, but also to reduce any losses that come from extra surface contact. We want to increase the throughput so that we could adequately sample the full range of complexity that we might be interested in sampling. We wanted to improve the reliability of our prep, and by preparing many cells in parallel, this allows us to reduce batch effects that come from samples being prepared on different days. And we want to automate the process further to take out any human error that could be associated in handling such small volumes. So to this end, we developed our method, Mpop, for nano-ProteOmic sample preparation, where we're using the cell-in-one liquid handling and cell dispensing system that you just heard about that has really enabled this prep to go swimmingly. And while this machine is really great and makes the sample prep really easy, a lot of the ideas we're going to be talking about today are kind of general and maybe applicable to another liquid handling robot or some way you also want to design the sample prep. And just to give you an overview of what our sample preparation looks like. So we're preparing our single cells and droplets of 5 to 30 nanoliters on a fluorocarbon open unpatterned glass slide. And here you can see a picture of the glass slide where each droplet would correspond to a single cell. And so on this glass slide at room temperature inside the cell in one, we first dispense our cells into a lysis solution. We then digest our proteins that have been denatured by the lysis buffer into peptides. We label our peptides for multiplex analysis, and then we pool our samples together and collect them and put them into mass spec inserts for downstream analysis by LCMS. And so now I'm going to be walking you guys through our Mpop protocol. And I'm really excited to talk about this method that we've developed that really is the most robust, a really robust and reliable and sensitive way to prepare up to over 1,500 single cells in a day. It's something that we're using all the time in our lab, and it really has become the workhorse for our sample preparation. You're going to see a ton of data at this conference that has been generated by the Cell in One, not only from my talk today, but also from Gray Huffman's talk on Wednesday, and where he's analyzing primary macrophage samples under a variety of different stimuli. And so yeah, let's start off. So first, what do you need to get started to perform Mpop? Just to summarize some of the materials, like I said, we're using the Cell in One liquid handling and cell dispensing system. I really can't say enough great things about this system. It's very robust. It works really reliably, and the cell dispensing capabilities have not only allowed our Mpop sample preparation method, but also works super well, as Nikolai mentioned, for sorting into 3D4 well plates. It's actually allowed us to get far and away the best data that we've had from the plate-based methods as well. And additionally, like I said, we're performing our reaction just on kind of your typical microscope slide that has a fluorocarbon coating to prevent any peptides from binding and to kind of give it a hydrophobic property that allows water droplets to sit nicely. And you can find these slides. They're commercially available and sold by Cyanion. We're using two different nozzles that they supply, one that's better for handling viscous protein solutions and organic reagents, and another for cell dispensing. We use a 3D4 plate, like Josh mentioned, to load our samples, and then any reagents that you might need for your proteomic sample prep, such as your lysis solution, your cells, your buffer, your trypsin, and labeling reagents. And so one thing I just want to talk about before you get started is the design layout for how you want to space your droplets. This is something that's really easy to program with the Cell in One. We're typically using 16-plex reagents with the isobaric carrier strategy, which allows us to put 12 single cells per mass spec set. So you can see on the left-hand side, we have some droplets kind of laid out in arrays of 12. But the system is really flexible, and we love our open glass slide design because this allows us to adjust on the fly if we're interested in doing different layouts. And you can see, in fact, here the other day we were preparing some samples that were 2-plexed, and here you can see it was really easy just to go in and change up that layout and adjust it for whatever needs you may have, whether you're interested in label-free analysis, whatever it may be. And this allows you to kind of optimize your space and increase your throughput as much as possible. So you have everything you need. You know how you want to conduct your experiment. The next step is to get set up. Here you can see on the left-hand side, we have 4 glass slides in place. And like I said, this allows us to prepare a little bit over 1,500 single cells simultaneously. And then you have your 3D4 plate on the right for picking up reagents. And so you have everything you need, your system set up. The first step in the process I'm going to talk about is cell lysis. And so this was actually not such a trivial problem for us to solve. We knew we wanted to get away from, we didn't want to use any detergents and reagents that weren't compatible with the mass spec, as those cleanups can introduce losses that make single cell analysis challenging. But additionally, we needed to also get away with the last lysis method that we developed, which was a freeze heat cycle, because like I said, we're carrying out these reactions at room temperature on this glass slide, and heating the sample to 90 degrees just wasn't an option, as it would cause our droplets to evaporate. So another lysis technique that we'd had some success with in our lab was using organic solvents. In particular, we'd use acetonitrile, and this had worked very well for us. So when we got started, this was the first thing we thought to try out. And kind of, naively, I thought, oh, what could go wrong? But we had a lot of problems along the way in handling the reagent that has really low viscosity. And additionally, once we kind of got those tweaks ironed out, what happened was the second we dispensed the sample on the plate, it instantaneously pretty much evaporated. And so it wasn't sticking around long enough to lyse our cells. And also we didn't have the option of kind of dropping our cells nicely into the lysis buffer, unless of course we wanted to, fill the humidifier that controls humidity with acetonitrile. But Nikolai cautioned me against that. It probably wasn't one of my best ideas. So what we did is we tried a variety of different organic solvents to see kind of what worked best with the dispensing and what would kind of have nice properties that made itself amenable for cell lysis. What we kind of finally landed on was Dmso, which has a really low vapor pressure and sticks around on the slides for a really long time. So it kind of had all these, and it was really easy to handle with the instrument. So it had all these nice properties. But to be honest, I'm not sure it's ever been used for cell lysis in this sense. And we really had no idea if this was going to work well. So we set out to do some experiments to see how well Dmso did at permeabilizing the membranes and making the proteins accessible for trypsin digestion. So I'm going to describe one quick experiment that we did to validate this. And we have done many more. And if you're interested, I can talk about that at the end. But just really simply here, we have some cells, some regular cells and some cells cultured with heavy arginine and lysine, where we're able to distinguish proteins coming from each culture and on the mass spec. And so we prepared one sample with a really standard 6 molar urea lysis, and then our other sample with 90% Dmso. And after incubating our samples for cell lysis, we then diluted the lysis buffers and combined them for trypsin digest and subsequent cleanup. And so what we saw from these results is that the protein extraction efficiency from the Dmso lysis was very comparable to the protein extraction for urea, and we further didn't see any particular bias coming from the various cell compartments. And so this gave us good confidence that this approach was viable to work on a single cell level. And so the first step is dispense small droplets of just 5 nanoliters of Dmso on various positions on the slide. Here you can see a video of the cell-in-one in action dispensing some of these Dmso droplets. And so after you've dispensed your droplets for cell lysis, the next step is cell isolation. And here you can see a video of the cell-in-one dispensing cells to our droplets. And this process goes pretty quickly. For us to dispense a little over 1,500 cells, it takes us a little bit under an hour. And we've even done this with some complicated samples that are originally sticky and we've had to filter. So we feel pretty confident that we can get the number of cells we'd like to be prepared, sorted in a reasonable time frame. And so after you have your cells dispensed and they're sitting not so comfy in the Dmso droplets, the next step is to kind of set up the humidity control that we need as we're eventually going to add aqueous to our droplets to prevent any unwanted evaporation as the samples are sitting on our glass slide. So there's a couple ways you can do this with the cell-in-one. There's humidity that you can, a humidifier that you can use to kind of control and keep the relative humidity high inside the instrument. And there's also a cooler that chills the plate to kind of the right temperature right below the dew point so your droplets stick around. And additionally, we further control the local humidity by dispensing an array of water droplets around our sample of interest, like you can see here. And this kind of gives us the ultimate control to prevent any of our droplets of interest from evaporating. And so the next step is to dispense our digestion buffer. The great thing about using really small sample volumes for cell digestion is that we can really increase the concentration of trypsin and in our samples to allow for really great digestion of our single cells, while also keeping the total amount of trypsin in our samples much lower than in our order of magnitude lower than in our plate-based prep. And someone just asked about the chromatograms. The effect of this is that the single cell sets, the peaks are really sharp and really don't look differentiable from our runs where we're running carrier only. And this is a really nice feature for our samples, for our sample prep that doesn't kind of translate as well when you have to add more reagents. Additionally, we're using heapies for our buffer, getting away from what we were previously using TEB, which is volatile and evaporates out of the sample. So we needed something that would stick around in solution and was more robust to evaporation. So after the sample's digested, we digest for 4 hours. The next step is to add our labeling reagents for multiplex analysis. So the first step in this process actually is to just let our peptides dry out on the slide. And so this is actually a really nice feature of our prep, as it allows for a pause point post-digestion, which I hadn't really thought about because I'm kind of used to getting in early and trying to make sure the prep goes smoothly and in a timely manner. But I was talking to one of our collaborators at Merck, David Perlman, who's expressed interest in getting the sample prep up and running in his lab. And he said, well, what if I start the prep in the middle of the day and I have a meeting or something and I don't have time to finish it all up. So this pause point, we think that the peptides are not reacting too much on the slide as they're dried out, and they can kind of stick around for a while and allow you to pick up the rest of the sample preparation later if need be. So we're to label our peptides, we first dissolve our TMT to 20 millimolar and 100% Dmso. So this is also a little bit atypical. You might be used to using acetonitrile for labeling your cells. But this is kind of something that we came up with that has really enabled us to have a robust workflow that has really allowed us to extend the throughput just past just a few samples. We had a lot of trouble handling pure acetonitrile, and the consistency and the robustness of dissolving the labels in Dmso has really made the throughput something that goes smoothly and has allowed us to really use this sample preparation method frequently. So after you've loaded all your labels into the plate, you aspirate each label and load the field that specifies which label goes to which cell, and then making sure to flush the remaining label in between each dispense to prevent any cross-labeling. And then we label for about an hour after the last label is dispensed. The next step after labeling is done is to quench the labeling reaction. Here we're dispensing 20 nanoliters of 5% hydroxylamine to quench the reaction twice. And so this is also a little bit atypical. And the reason for this is that one really important control that we include in our experiments is a negative control, which receives all the same reagents as a single cell, but just does not receive a single cell in the droplet. And so here you can see the negative control on the right, and we found that kind of a major source of background in some of our sample preparation was due to cross-labeling that kind of added an extra unwanted layer of signal. And here you can see very low to almost no signal in our negative control, which gives us confidence that the signal that we're measuring in our single cells is truly coming from our single cells, which is great to see. So after you've labeled your samples, you have the sample pickup. And so for a while, we've been doing this by kind of pipetting water onto the slide to pick up our samples and then dispensing acetonitrile. Fortunately, I was able to work with Josh yesterday to kind of automate the very last part of this. And so now we're able to pick up these samples with the cell in one, and I'm gonna just kind of let this video play it. It pretty much speaks for itself. So here we're dispensing acetonitrile onto the slide to recover the peptides. We're picking up the sample, and then we wait. We dispense some of the acetonitrile back out onto the slide to ensure proper mixing. We let it sit for a while to assure that all the peptides are totally solubilized. And then we aspirate the sample and pick it up the plate, and you can kind of watch that happening here. And so this is something we're really excited about. So after this, the next step is to just take the contents of the PDC and dispense it right into a 384 well plate that's kind of positioned on the right-hand side. And so this is also a really awesome feature. As most auto samplers, you can kind of just plug in the 384 well plate, and with not too much effort, I think it's built into most auto samplers that they're able to do pickups out of a 384 well plate. So this is really nice. It's a way for us to kind of finally automate the last bit of our protocol and dispense the samples into something that's already with kind of no real additional outfitting compatible with most auto samplers. So this was kind of a really nice last feature to add, and thanks to Josh for his expertise and know-how that kind of made this possible. So just to summarize some logistics, our prep takes about 8 to 9 hours to prepare over 1,500 single cells. And this involves about 2 to 3 hours of hands-on time. And like Nikolai said, this prep, some of these steps might have looked complicated, but I've really gone out of my way to make sure that all the information is available for anyone to be able to pick up this prep. And in fact, a couple other students in our lab have been able to pick up this prep and within just a few days kind of complete it with everything they've needed. And additionally, along these lines, I have a protocols.io that goes into much more detail than I went into today for anyone who's fortunate enough to have access to a cell-in-one and interested in preparing samples via Mpop. You can find that on the Mpop website that you can kind of just Google Mpop Slavov. It'll be one of the first things that comes up. And also you can comment on this protocol. And so if there's anything that's unclear, it's kind of, I'm going to be continuously updating it. People have already been commenting, giving me feedback to how to improve it. And so that's really awesome. So please, if you decide to check it out, feel free to remark on anything that's unclear. So you've seen this method that we've told you can analyze many single cells in parallel in one day. And your next question naturally might be, okay, so how does the data look from the sample preparation method? And so that's what I'm going to be talking about next. Our model system that we've been working with is just some cell lines that we had on hand in our lab. Here you're seeing some U937 monocytes and some HeLa cells. And when we when we project our data into a low dimensional space, we're really easily able to separate cells based off their proteomes. Here you can see kind of the HeLa cells and the monocytes clustering very well on across PC1. But actually, we don't love using PCA as a way to totally validate our single cell measurements because, as many people know, and as they saw in the early days of single cell RNA sequencing, this separation can be driven by a number of technical factors: missing data, cell size, and a number of things that might mean, okay, you have some separation, but are you really measuring biological signal? So the next thing we like to do is we like to correlate our single cell measurements to bulk measurements. And to do this, what we do is we just kind of average the protein abundances across our single cells and our monocytes and our HeLa cells. And we compare that protein ratio in our single cells to the ratio of some HeLa cells and monocytes analyzed in bulk methods where we have much higher degree of confidence in the accuracy of the quantitation. And what you're seeing here in the plot on the right is that we get very good concordance between our single cell measurements and our bulk samples. And this kind of gave us an extra layer of confidence that we're doing accurate measurements on the single cell level. And so you probably didn't need to do single cell mass spec analysis to tell you that HeLa cells and monocytes are quite different. You can know that just from looking in a microscope. So a much more interesting question is, could we detect any meaningful heterogeneity within one of these cell types? And like I mentioned earlier, one of the ways that a clonal population of cells might differ is just based off their cell cycle state. So first, we kind of did a pilot experiment to see if it was feasible to differentiate cells based off their based off the cell cycle. So what we did here is we pre-sorted cells into 3 populations based off their DNA content. So we have cells with one set of DNA in G1, and then cells that are replicating their chromosomes in S phase, kind of in the middle part of this intensity DNA content graph. And then the last peak on the end is cells in G2 phase where they fully replicated their DNA. So we have sorted populations of cells where we have a little bit of ground truth roughly about their place in the cell cycle, and we're able to do single cell analysis on these samples. And so what you can see from the PCA here is that we're able to roughly separate cells based off their cell type. And you'll also see that this doesn't explain a tremendous amount of variance across PC1, which is maybe to be expected because we don't think that every protein in the cell is regulated with cell cycle. So what we can do further to kind of further look at this is we can take a handful of proteins that we more confidently know to be involved in cell division. And so when we further analyze the PCA based off these data, you see that a much higher degree of variance in the data is explained by state in the cell cycle. You can see that the G1 and G2 cells cluster kind of very far apart on the sides and with kind of some cells in S phase in the middle as the cells transition throughout the cell cycle. But also, as I mentioned earlier, we don't love using PCA in and of itself as a validation of our measurements. So to look at this a bit further, we did some Go term enrichment based off these different clusters, and we saw a couple things that we expected, such as regulation of the cell cycle and microtubule organization upregulated in G2 phase as the cell is getting ready to divide and become 2 cells. But additionally, we didn't want to analyze all of our samples this way, as you might be interested in analyzing the cell cycle in a complex biological system where you probably don't have the luxury of really nicely sorting out populations based off DNA content and having confidence that these populations are homogeneously somewhere in G1. So we set out to do this analysis on our more unsynchronized data. And so what we did is we projected both our HeLa cells and our monocytes into a low dimensional space based off a handful of cell cycle markers. And then we shaded the PCA based off the abundance of proteins associated with various degrees of the cell cycle. And what you can see is for this handful of cell cycle regulated proteins, we can see that we have separation based off our principal component analysis that's related to the abundance of proteins in the cell cycle. And additionally, we see that this behavior seems to be pretty similar regardless of whether the cell was a HeLa cell or a monocyte. But the nice thing about doing single cell mass spectrometry, again, is that we didn't just measure a handful of proteins that we think are involved in cell cycle regulation. We were able to take measurements of many proteins. So you can kind of ask, going back to this picture that we talked about earlier, if you're interested in figuring out what else in the proteome might be going on other than just a handful of proteins, we can start to look at this. And so to do this, what we did is we took the handful of proteins in various Go terms. Here you see microtubule-based processes, and we looked at their degree of covariation with proteins from known cell cycle phases. And here what we're looking at is the correlations plotted for the proteins in this Go term with markers for various different phases of the cell cycle. And so you can see that this Go term behaves similarly for the HeLa cells and the monocytes, and we found many such similar Go terms that that were correlated, significantly correlated with our markers for the cell cycle. And rather than show you 100 box plots, we chose to condense the rest of that information based off taking the median values of the correlations and condensing that information in a heat map. And you can see that there are many such Go terms that associate with our cell cycle markers that are similar between our monocytes and our HeLa cells. And similarly, we were also interested in looking at ways in which there were differences between the cell cycle for the HeLa cells and the monocytes. And so we were also able to find some functional groups of proteins that correlated differentially between the two cell types as a cell with the cell cycle markers that we measured. So in conclusion of what we talked about today, we have a method that allows you to, in parallel, analyze over 1,500 single cells, prepare over 1,500 single cells in one day. And additionally, we found functional groups of proteins that covary with the cell cycle, getting at that original question that we talked about of unpicking complexity in our samples and the need for doing single cell measurements. And so that's my talk for today. I just want to say thank you to all the people who made this possible. Professor Slavov, as he mentioned in the beginning of the talk, for giving me a chance to work on this really cool research, and to my co-author, Gray Huffman, and also Josh and Aileen, who have been two engineers at Cell & One, who have provided tons of support and have been there every step of the way that we needed them. And also thanks to the funders for making this possible. And I just have some of my contact information down at the bottom. I love to talk about this stuff, and I love interacting with people. So please reach out if you're interested in Mpop, or if you're interested in any of the data that we've been generating, or you just like to chat about single cell proteomics. Thank you.
Thank you. So we have a few questions for Andrew from the chat, but this time, just to alternate, I'll first start with questions from the audience, if there are any. John at the back.
How long do you analyze those 1,500 cells?
Yeah, so, yeah, that's a great question. So, okay, so yeah, we have 1,500 single cells prepared. We're running our gradients are about an hour and a half long. So this is actually over 10 days worth of mass spec time if you were just continuously running. So kind of traditionally, we had been limited in our lab. Theoretically, we were limited by mass spec time, but we were finding that in practice, we actually couldn't generate enough samples to constantly keep our mass specs busy with single cell sets. So this is kind of another positive step in that direction of constantly keeping our mass specs busy, analyzing the single cell samples of interest.
Yeah, the fact sorting was something that always was a big challenge for us. And the cell isolation in Cell & One works day in and day out, like the sorting. Okay, so we have quite a few questions from our virtual audience. And the first one is, what is the final concentration of peptides that you're injecting into the mass spectrometer? So it's about a microliter. Yeah. So our sample from 12 single cells is P pulled into a 1 microliter that's being picked up by the autosampler. And this is in no way a limit. We can reduce that volume. And in fact, we're working on reducing it, especially now with the automated pickup.
Yeah. Yep.
Have you tried RapidJest as a denaturant? If yes, how did it compare to Dmso?
I've thought about trying RapidJest. I actually have some sitting in the fridge 4 feet behind the cell in one that I work with. The sample prep has been going well, and we haven't really seen any evidence that we need to improve the lysis further. But it has been something that I thought about looking at, but we've been busy and just haven't had time to try everything out.
Dmso works so well, and it's completely clean. We can evaporate it, and there's nothing left on the column. And I prefer nothing left on the column than something that's supposedly mass-spec compatible. Can Dmso dissolve cell walls in plant cells?
So I've mostly been focused on mammalian single-cell proteomics, though I'm not sure what the answer to that question is. I just would like to point out as kind of a feature of being able to work with these really low sample volumes is that you probably could get away with using a little bit harsher lysis reagents if you needed to, just because the volumes are so small that say you wanted to just analyze one plant cell at a time. Even if you had a fairly high concentration of some more heavy-duty lysis buffer, you might find that this is still mass-spec compatible. So yeah, that's a good answer.
So we have a very nice comment from John Yates. He says the sample pickup strategy is a genius.
I can't take all the credit for that. So
we had a celebration when we got.
We did have a celebration yesterday. It was nice.
Can you use Dmso to lyse bacteria cells? I think that goes in the same category as with plant cells. You would certainly denature proteins and dissolve all the membranes. The cell wall, we don't have experience as to whether that can be used. Perhaps one can also add an enzyme to first predigest the cell wall, and then you can certainly use Dmso. Dmso would denature all proteins and dissolve all membranes. Amazing. I've been trying to do that for months, wasn't able to get those results. Well, get in touch with Andrew.
Please.
And I'm sure he's going to help you.
In no time.
Do that. Will HIPAA be tolerated well by LC-MS systems? Did you try TEP?
Yeah, so this is a great question. This was more of originally a theoretical consideration that we didn't want to include TEB because we thought it would be volatile and potentially evaporate out of our samples and cause the pH to not be so well sustained. We see no negative effects of the HIPAAs in our solution. It's at even a pretty low concentration in our droplets, and those droplets are diluted nearly 100-fold when we're injecting them into the mass spec. So the concentration of HIPAAs is very low, and we really see no negative effects of ionization. And we have many controlled experiments for this, is just running a sample without single cells and then with single cells post, and we do not see any degradation in the total ion current.
Exactly. So we don't know that TEB cannot work. Maybe you can make TEB work if you're interested in that. We simply saw no need to try doing it, and we thought that HIPAAs would be much more robust. As Andrew explained, it's a tiny amount. And another consideration to add here is that the way we run our LC-MS is that everything that elutes before the peptides doesn't get into the mass spec. And that's when the HIPAAs is going to elute. So you can look up the description for that in our protocols. And empirically, what we see practically is that we can run the mass spec continuously in the course of weeks without substantial contamination or problems. What's your labeling efficiency like for TMT when you dissolve in Dmso?
That's a great question. So that was another thing that we weren't totally sure was going to work really well. We don't need to, I guess, dry out our samples and label in 100% organic like we do, but we thought this would be a really nice feature of the sample preparation reaction. So we did a lot of controlled experiments, dispensing digest on the slide and letting it dry out and labeling it, doing it in a PCR tube. And I've never seen the labeling efficiency be anything worse than 99.9% or whatever you'd expect from your normal acetonitrile-based protocols. I do want to mention that while we've had a lot of success doing this with TMT, a ton of success, also tried other labeling reagents where we've not seen that this transfers so well. So this is something that if you want to incorporate it in the exact same way into your protocol, but with a different labeling reagent, you might want to test out that, as different labeling reagents will have different chemical properties and different solubilities.
Is it possible to estimate copy numbers in single cells, such as in HeLa cells, any cells that have more ploidy?
copy numbers of proteins?
So the question is not clear for that. Initially, it seems to be more interested in ploidy because there are chromosomal abnormalities. Yeah. In principle, one should be able to see that. In practice, we haven't tried. Our data are publicly deposited. So if you're interested in doing that, please go ahead and let us know. Should be possible, but we didn't try doing that. How long did it take to prepare the single cells used in the data set?
Yeah, so maybe just a little bit anecdotally, when I first kind of developed the sample preparation and what we have so far available in our preprint, we were preparing just kind of 8 sets at a time. And so we finally got things working out for kind of a small set of samples. And the next question was, well, okay, now that things seem to be working, let's drive the throughput. So we kind of eventually we stepped it up to 12 sets. And then we were like, well, this still isn't much of a benefit. So let's just try to see how many we can squeeze into a slide. And our next iteration was kind of preparing 48 sets in a day, which is around 500 single cells. And so the data that I showed today is from 2 different experimental days where we prepared 500 single cells in parallel. And now we've even pushed that further, and we've totally cramped out all the space that we possibly can on these slides, and we're regularly preparing 1,500 with essentially no issues. But the data we looked at today was prepared over the course of 2 different days.
And that also speaks to another benefit of the sample prep, that there isn't much batch effect, certainly less than what we've seen with other methods. I think that there might be room to further improve it, but at this point, the sample prep for us is really not limiting. We can keep multiple instruments fully busy with just a single cell in one instrument. Great talk. How many proteins you were able to detect per single cell?
So in the PCA, we were showing there was 3,000 proteins quantified across all single cells. The number per single cell in the experiments we did is somewhere around 800 to 900 per single cell. This is a function of a lot of different things. I would say, personally, my thoughts are that it's much more a function of how you're running your LCMS and your acquisition strategies and the sensitivity of your ionization and everything than it really is prospect of the sample preparation. But one of the great things about the Mpop sample prep is that the samples, even on the single cell level, are really clean, and the low level of contaminants kind of allows you to detect as many proteins reliably as you could imagine detecting in with any kind of clean sample that you're interested in. And the level of missing data in the single cells is pretty low. And so really it's the question of how many peptides you can detect.
So in terms of coverage, what Andrew said is spot on. Just 2 things to add. One is that because of the cleaner spectra, we are able to assign a confidence sequence to over 35% of the Ms2 spectra, which is substantially higher than what we've been able to do previously. The other thing is, if one is interested in just increasing the depth of coverage, one can easily extend the LCMS gradient. We keep the active gradient short because we want to be able to analyze large number of single cells. And that's a question of an experimental trade-off, depending on the preference of experimenter. Do you prefer throughput, or do you want to have 2 cells with deep coverage?
Now that you're pushing like significant numbers of cells, 1,500, and I know this is a issue all across the board for fact sorting, how much do you worry about what biology is going on in the cell before you add the Dmso, especially for the experiment you just talked about?
No, that's a really great question. So one of the nice things about proteins is they're a little bit less finicky than RNAs. So it takes us about an hour to dispense all of the single cells. So they're obviously sitting on PBS. We have been meaning to, for the longest time, do controlled experiments to have cells sitting here in a 1x PBS, and then taking another population an hour later and looking at the differences between the proteomes. We haven't gotten around to doing that, despite clearly having, we should have. But what I can say is that we really see no bias in our single cell data that indicates any differences between the cells dispensed at the end versus the cells dispensed at the beginning. But this could be certainly done by more controlled experiments that I assure you we will eventually do once we get around to it.
So I quite like the next question. It's about quality controls. What kind of quality controls do you use? I think quality controls are super important. I think one shouldn't even think of doing single cell mass spectrometry without building in a lot of positive and negative quality controls?
Yeah, so I'll talk about this in a couple of ways. On the sample preparation side of things, kind of like I talked about in my presentation, one of the biggest checks for us is the negative control, which essentially receives all of the same reagents that a single cell would, but just without any single cell. And so what we see from our Mpop sample preparation is that the signal in our negative control is really low, and this is in part due to the small amount of reagents that we're able to add. We see in many cases sometimes with the plate-based prep, especially in sets that have been sorted with a fax, we see really high, sometimes, or potentially sometimes we can see higher background in our negative controls. And so this is kind of a nice feature. There's also a lot of important controls that that should be added on the data analysis side of things. Like I mentioned, just totally using PCA separation to validate your measurements is probably not the best approach, as it could be potentially technical artifacts. So we really like incorporating in all of our analyses correlations to bulk samples that we have more confidence in their level of characterization.
Want to mention about DMS?
Oh, and yeah, that's a great point. And we have the software. You can see a paper published by Huffman a couple of years back that we use kind of as an initial quality control to check all of our samples. This is a really nice, it's a R Shiny app, and you can look at tons of stuff right off the bat, like, contamination in your samples, single cell statistics. There's a lot of cool single cell statistics that we have in there that allow us to assess how well the prep went, such as the digestion of single cells, which is kind of a really interesting problem, since the identifications are driven by how well your carrier sample. So you're not really able to look at the rate of miscleavage in the single cells conventionally with the carrier approach. But what we can do is we can kind of compare the ratio of miscleaved to non-miscleaved peptides in our carrier and miscleaved to non-miscleaved peptides in our single cells. And so we kind of benchmark our single cell digestion versus our carrier digestion. And so this has been a really useful metric for us. And we see that the samples are pretty much digested as well. And definitely check out DMS. There's tons of great plots in there. I don't have time to talk about all of them, but
a huge time saver. All of the metrics are generated in a completely automated fashion. Have you analyzed the effect of co-isolation? Do you have any measurements to speak of co-isolation?
Yeah, that's a good question. I mean, just as kind of everyone looks at, we measure the PIF of our samples, and when we're doing our analyses, we filter on a certain degree of spectral purity. But this isn't obviously a perfect way to assess the amount of co-isolation. If you're using a top 7 method, by the time you're on your 7th fill time of, say, 300 milliseconds, things could have shifted at the MS1 level. You could potentially have another co-isolating peptide. What I can say is that single cells may be prone to the effects of co-isolation in theory more than potentially another sample. But again, we validate this on the bulk level by comparing our samples to more traditionally prepared bulk sets. And we see that the correlations generally align well. And this speaks to kind of the amount of noise introduced by co-isolation.
So our main strategy to minimize the effect of co-isolation is to optimize the sampling of the elution peak apices. We use DMS for that, and that has been quite helpful. And then we further validate that the precursor ion fraction for the main precursor analyzing is high. We look at these distributions and remove all peptides that don't have very high precursor ion fraction. And
one last thing to say, I guess, so if you have a sample, so the nice thing about adding less reagents to our sample is that we have less things that are eluting over the course of our gradient that have tons of signal from our single cells. So, you could imagine that it would be much more problematic for some trypsin autocleavage product to co-elute because you have, more trypsin in your single cells potentially than another end, like co-eluting another peptide that
the limited co-isolation that we see in our data to some extent is the silver lining of not having very deep coverage. Co-isolation is always a much bigger problem in any kind of experiment and data set for lowly abundant peptides. So to the extent that we are still looking at relatively abundant proteins, they're less affected by co-isolation. Is there a sheath liquid in which the single cells are sorted?
We sort the single cells in 1x PBS, and the cell one's really great. It dispenses them in volumes of just 300 picoliters. So we see no negative effects from kind of the sheath liquid. But generally, the system just operates with your average DI or clean water, kind of. There's a filtration system. And yeah, so the system just operates off purified water.
We have seen in our experience negative effects of sheath fluid being present with fact sorting, but not with cell-in-one. And this all has to do with the cell-in-one droplets being much smaller than the fact droplets. How often do you have to change the nanocolumns on liquid chromatography with single cell samples? Is there a difference compared to general proteomics samples, which mostly undergo rigorous desalting step?
Yeah, we are able to keep our columns running for months pretty much, and we've recently really scaled up the amount of single cell samples we're analyzing. We've ran recently, Gray I think ran 60 sets in a row sequentially, and we didn't notice any significant degradation in chromatography or ionization. So that's a little bit I can speak to it. And yeah, our columns have held up generally pretty well. I guess shout out to ion optics and pharmafluidics.
And the very small volumes that Andrew is using for the sample prep really contributes to having very little extra chemicals being added other than the peptides. That's a major advantage of the small volumes. Great talk, amazing results, very inspiring. I like this person.
Thank you.
May I know how do you set up the mass spec? What is the total volume inject in the mass spec and the gradient? Do you use a trap, and what is the column ID that you use?
Sure. We do not use a trap. The column ID is 75 microns. And yeah, our injection, so our autosampler, our loop is set up for 1 microliter pickup. We actually have to load about 1.4 microliters into our current vials to make sure that we don't end up picking up air. I'm actually excited to switch to the 384 well-played pickup because this is going to allow us to optimize this a little bit more. But in general, part of it is kind of what your autosampler can do. We've run samples on a different autosampler that could have variable inject amounts. And I said, well, why not just load up 5 microliters and then take 5? Well, that'll allow you to kind of obviate any risk of air injection and stuff like that. What else was the question? The gradient. And we run, for these samples, we are using about 300 millisecond Ms2 injection times. Our gradient is standard. We go from about 4% buffer B acetonitrile to 35%. And yeah, the one parameter that we've kind of fiddled with the most is kind of the Ms2 fill times. And this really depends on how big the cells you're analyzing are. This is like a massive factor. Obviously, HeLa cells are almost 5 times the size of maybe, for example, the mouse macrophage primary samples that Gray is going to tell you about later this week. So you need to change your fill times depending on what kind of samples you're analyzing, and to some extent, personal preference for the data completeness you would like to have in your samples.
Chromatography in our experience is absolutely key, and our philosophy on principle has always been to use commercial solutions, because I would like what we use to be available to anybody else. So we have found a couple of commercial solutions that work well in our hands, and that includes ion optics, and the pharmafluidics columns also perform well. Both of those are good. Certainly important thing to optimize and keep an eye on when doing ultrasensitive analysis. Chromatography matters. If you had to pick 2-3 parameters that would take single cell protein measurements to over 3,000 proteins per cell, what would that be based on your intuition? So before I let you answer, I'll just preface this by saying that I'll spend a big chunk of my talk tomorrow discussing that. But I'll let Andrew.
Sure. Yeah, I think much more so than this being related to the sample preparation, kind of like I said, a lot of this has to do with how you're operating the mass spec. And we have a lot of really interesting ideas how to increase the number of proteins that we're quantifying per single cell that you will be hearing about in the near future. So
one of the most trivial ways is just extend the active gradient. Of course, that's going to reduce throughput, but that's a very easy way to immediately increase the throughput. Another one is just decrease accumulation times, which of course is going to result in sampling fewer copies from the single cells and therefore noisier quantification. So it's very difficult to characterize the performance of single cell proteomics with a single number. And that number is certainly not proteins per single cell, because we could easily just tweak the parameters of longer active gradient, shorter fill times, and get to the 3,000 proteins in a way that it's not going to give us 3,012 quantified proteins. And then the question, how do you get to 3,012 quantified proteins? It's a great question that we'll have a lot more to talk about and things to say. I'll just mention that one of the approaches that already is being demonstrated in practice without clear trade-offs is to use more intelligent data acquisition, and in particular focus the instrument time on precursors that are identifiable instead of just messing anything that is abundant, which might be a trypsin autoclave product or something that is not identifiable. And both Chris Rose and Ray Huffman are going to talk potentially about such approaches. How about number of ribosomal proteins? They're all okay. There are always a lot of them in normal proteomics. So yeah, we
identify lots of ribosomal proteins. We spent some time doing some analysis looking at the correlations between different ribosomal proteins to see to try and take a step towards analyzing complexes from the bottom-up approach. And we see that we're able to look at the correlations between a wide array of different ribosomal proteins, and so things like that. I don't have a great hard number, but maybe on the order of, I don't know, 30 or so, maybe more.
Have you tried extracting and processing the cells from frozen samples?
I have tried extracting and processing the cells from frozen samples. Actually, the data that you saw today was from samples that were previously frozen down in FBS and 10% Dmso, as you would normally freeze down cells for cell culture, and then brought up the day of and washed off with 1x PBS, and then we loaded them in normally. So yeah, this works really well in our hands. We haven't seen any negative effects of it, but I certainly don't have super well controlled experiments to tell you about what shifts in protein abundance may be happening. But you could also say, if you're treating all the cells similarly, hopefully, and you're taking relative measurements, hopefully this isn't much of a problem.
What's the cost per cell now with all the costs involved, equipment, etc., but not MS?
Great question. So a really cool thing about using the small volumes as well is we're able to add a lot less reagents, especially, TMT is kind of one of the more costly reagents. And essentially we're able to prepare with the Cell-in-One essentially 7 plates worth of samples with the TMT that we would normally use for 1 plate's worth of samples. So I don't have any exact numbers for you.
So the cost is completely dominated by LC-MS time. This is by far the most expensive component. Now with sample prep, it depends how you estimate that. Cell-in-One is cheaper than a fax sorter, as we already said, but of course you need to have access to that. And whether you're going to factor this as a facility user or as owner of the instrument and how you compute the cost. Generally, the cost of preparing a single cell is below $1 if you're paying facility fees and if you're doing this in a high throughput manner. And the cost of the LC-MS time is really what's pricey.
You want to send me your samples, we can talk about the rest offline.
So a question recycled from previous talk. What are the considerations about cell density or concentration of the starting material taken for single cell sorting?
Yeah, so that's just really a great feature of the Cell-in-One. Like Josh said, if you have a really limited amount of cells, you can even just get the ones that you don't collect, you can dispense into a collection tube that you can even rerun. But it's generally rerun at about 200 to 300 cells per microliter. And the efficiency is pretty good. About half the cells that go through if the sample is kind of well taken care of get dispensed. So anecdotally, we had some macrophages that we were once analyzing that were on some really nasty fibronectin that we could only get off like, I don't know, somewhere in the order of 1,000 cells. And we were able to analyze like a couple hundred, I'm pretty sure. So it's pretty good on that end. Credit to the Cell-in-One mostly.
Does the cells have to remain viable for selection? If so, how do you check viability?
That's a good question.
One can use all the stains that you would normally use with fact sorting, such as trypan blue or propidium iodide that can be used with cell-in-one as well. And we haven't seen, at least with the cells that we have used, we haven't seen those cells losing viability and rupturing. What are the acquisition modes compatible with your single-cell proteomics workflow? DDA, DA?
Yeah, good question. We've analyzed single cells, mostly DDA, as the multiplexing of the labeling allows us to generate a large number of samples, as we kind of talked about in this presentation, to address some of those interesting degrees of complexity that you might find in your samples. But I'll tell you, we also definitely have analyzed single cells with DIA, I won't say more than that.
So the final question is a perfect segue for our next talk, and we're just on time. The final question is whether real-time search has helped to increase the depth of protein coverage.
So I actually don't have a ton to say about that.
So real-time search, I'll just talk a little bit more in the category. Real-time search is essentially intelligent data acquisition. And intelligent data acquisition has helped a lot increase our coverage by a factor of 2. And Gray Huffman is going to tell you all about that on Wednesday. But before Gray talks about it, he's used a tool, MaxQuantLive. I'm going to tell you about the person who initially developed it and who helped us adapt it to single-cell protein analysis. So with that, I'll just thank Anshu for this presentation. And I would like to encourage those of you in the audience here present in person and those of you who are joining us virtually to ask questions from Anshu. If you try to use nPOP and you're having trouble, get Anshu to help you.
Yeah, please. The method won't be impactful unless I can get all of you, if you're fortunate enough to have access, kind of up and running. So, yep.